So what is CRISPR gene editing? The basics. CRISPR is one of the most powerful scientific tools ever discovered. At its core, it's a genetic editing system, a way to cut, alter, or rewrite DNA, the code of life itself. Think of DNA like a massive instruction manual written in 4 chemical letters, A, T, C, and G. Until recently, humans could only read those instructions, but CRISPR allows us to pick up the pen and start rewriting them. But what makes CRISPR different from older genetic tools is its precision and affordability. Before CRISPR, editing genes was slow, clumsy, and expensive. CRISPR changed the game. Suddenly, editing a gene in a lab costs less than a nice dinner, and scientists everywhere can do it. How it works. At the heart of CRISPR is a protein called Cas9, guided by a custom strand of RNA. Imagine Cas9 as a pair of molecular scissors and the guide RNA as its GPS system. The RNA is programmed to recognize a specific DNA sequence, and when it finds its match, Cas9 slices the DNA at that exact spot. Once the DNA is cut, the cell scrambles to repair itself. Scientists can then slip in new instructions, adding, deleting, or rewriting genes. This means researchers can turn genes on or off like switches, fix mutations that cause disease, or even insert new traits entirely. Here's an analogy. Think of a 3 billion letter long book filled with typos. CRISPR is like having a search and replace function for biology, letting scientists go straight to the error and change it with unbelievable accuracy. Already, CRISPR has been used in experiments to modify crops, to resist pests, create mosquitoes that can't spread malaria, and even attempt to correct genetic diseases in human embryos. History/origins. The story of CRISPR begins not in a genetics lab, but in the DNA of bacteria. Back in the 1980s, scientists noticed strange repeating sequences in bacterial genomes, like a pattern no one could explain. For years, these sequences were a mystery, filed away under weird genetic junk. But by the early 2000s, researchers realized the truth. These repeats were part of an ancient bacterial immune system. When viruses attack bacteria, the bacteria store little mugshots of the viral DNA in these CRISPR sequences. Later, they use those mugshots with Cas proteins to recognize and destroy the invaders if they return. Then came the breakthrough moment. In 2012, Jennifer Doudna and Emmanuel Charpentier discovered that this bacterial system could be repurposed as a universal gene editing tool. It was simple, elegant, and incredibly powerful. They published their work, and the scientific world exploded with possibility. Just a few years later, labs around the globe were wielding CRISPR like a scalpel for the code of life. Doudna and Charpentier would go on to win the 2020 Nobel Prize in Chemistry, marking CRISPR as one of the most revolutionary scientific discoveries of our time. Impact/legacy. The potential of CRISPR is staggering. In medicine, scientists are testing ways to cure genetic diseases like sickle cell anemia, muscular dystrophy, and even some cancers. Instead of just treating symptoms, CRISPR aims to fix the root cause by repairing the faulty genes themselves. In agriculture, CRISPR promises crops that can survive droughts, resist pests without pesticides, and grow more efficiently to feed a booming global population. Imagine bananas that never go extinct, wheat immune to fungi, or tomatoes enriched with vitamins. But CRISPR's power also raises deep ethical questions. Should we use it to enhance humans, making people stronger, faster, or smarter? What if parents could choose the height, eye color, or even intelligence of their children? Where do we draw the line between curing disease and designing humanity? In 2018, a Chinese scientist named He Jiankui shocked the world by announcing he had edited the genes of twin babies to make them resistant to HIV. The backlash was immediate. and he was condemned and imprisoned for unethical experimentation. His case stands as a warning. Just because we can edit life doesn't mean we should, at least not without serious caution. CRISPR's legacy is still being written, but one thing is certain. It has given humanity unprecedented control over biology. Random facts that don't seem real. A CRISPR kit can be bought online by anyone for less than $200. Yes, even high school students can gene edit bacteria in their kitchen. CRISPR is so precise that scientists have edited genes in living organisms ranging from mice to monkeys, and in some cases have cured diseases in lab animals entirely. It's been used to engineer mushrooms that never turn brown, potentially eliminating food waste. NASA is studying CRISPR for use in space, in case astronauts on Mars ever need to genetically engineer microbes to help them survive. Scientists are even exploring CRISPR to resurrect extinct species, like the woolly mammoth, by editing elephant DNA. Conclusion. So, what is CRISPR gene editing? It's not just a tool, but a turning point in human history. For the first time, life's code is no longer untouchable, it's editable. CRISPR holds the promise to cure disease, end hunger, and reshape our world. But it also forces us to confront what it means to play God with nature. If you also like learning random things, subscribe. You have found your happy place. Poem, the scissors of creation. In the script of stars and strands we carve lines of fate once fixed, now bent and starved. Hands of mortals wield the quill of genes, rewriting destinies, unseen machines. Hope or hubris, we walk the divide, cutting tomorrow with the blade inside.